Welcome everyone, and thank you for joining us today for our webinar titled Understanding Ventricular Pressure Volume Calibration and Experimental Design. This is Andy Henton from Inside Scientific, and I will be your host for today's event. Our session is sponsored by AD Instruments and will be an in-depth exploration of conductance pressure volume loop science, including calibration theory and best practices, considerations for experimental design, and most importantly, how these choices can affect your resulting pressure volume data. We are joined by Dr. Jim Georgakopoulos, Chief Scientific Officer at Sunshine Heart Incorporated. Jim started his PV loop research in 1994 alongside David Cass. Over the next 10 years, together they worked on many firsts pertaining to PV loop science, including development of the first systems and methodologies for obtaining PV loops in the mouse, and were the first to publish a paper showing PV loops collected in an animal model of this small size. They went on to study force-frequency relations in the mouse, assessing calcium dynamics in vivo, and also study the role of nitric oxide in neural control of the circulation through the baroreflex. Following, Jim went on to join CVRx, which at the time was developing a device to electrically stimulate the baroreflex for the treatment of hypertension and heart failure. In this position, he applied PV loops to study the effects of the device and published the first human PV loop recordings, along with electrical stimulation of the baroreflex. In his current position, he is engaged in the development of a device targeted to assist the failing heart in advanced heart failure patients. Specifically, the device consists of a cuff which is wrapped around the ascending aorta, providing chronic counterpulsation. Interestingly, they believe that some of the effects of mechanically compressing the aorta may be due to aortic baroreceptor stimulation. Therefore, they are employing PV loops to separate the effects on the heart and coronary circulation from the peripheral vasculature.
Well, I'd like to welcome everybody and thank you for attending this webinar. As Andy mentioned, there will be a question and answer period at the end. Perhaps share my secret recipes of doing some PV loops, particularly in mice over the last 15 or 20 years. So the title of today's talk is Understanding Ventricular Pressure Volume Calibration and Experimental Design. We're going to briefly touch on introduction to pressure volume loops, starting with the end in mind, meaning what is your experimental design before you even start collecting your data and calibrating your data. And then we're going to go into the particular calibrations of the conductance catheter, especially stroke volume and parallel conductance or parallel volume, and wrap it up with just a brief discussion on once you have calibrated values, how do you put it all together. So as most of you are aware, since you are participating in this webinar, the ability to derive pressure volume loops is probably the most powerful methodology you can use to define all intrinsic cardiovascular conditions, coupling of the heart to the vasculature, separation of heart versus peripheral effects. It gives you really an entire picture of what the cardiovascular system is doing at any particular time. The other advantage also is you've heard of the collection of load-independent data, particularly one classic measure known as the end-systolic pressure-volume relationship, which you can see here is the red line connecting the upper left corner to the PV loops. It's important to understand that when you're collecting so-called load-independent data, the ability to vary preload and collect multiple pressure-volume loops instantaneously is one of the real advantages of using the conductance catheter to do this. It's really the only methodology that allows you to collect multiple beats and see them instantaneously. The other advantage of the catheter for those using larger animals and you have multiple segments is it's actually a very powerful tool to assess the synchrony from base to apex, and this has been applied in many clinical studies of CRT. And finally, the other big advantage of using the conductance catheter and pressure volume loops is it really provides the gold standard for assessing diastolic function through what's known as the end diastolic pressure volume relationship, and that's the blue line in the bottom portion of the PV loops. Again, collected over multiple beats to describe that full relation. And once you really have those two lines, the end systolic pressure volume relationship and the end diastolic pressure volume relationship, you really have defined the extremes, the lower and upper extremes of where the heart is operating for a given contractile function. So brief introduction. So how do we collect? Pressure is relatively easy to measure with multiple systems, but volume has always been the challenge and has really limited the application of obtaining pressure volume loops. As previously mentioned, the conductance catheter has really emerged as the sole method to provide instantaneous pressure volume data. And it essentially consists of 4 or more electrodes that you can see there in the blue color. The outer electrodes provide a current. They inject a current into the left ventricle, and the inner pair of electrodes are used to measure the conductance or the voltage drop across those 2 electrodes. And essentially, it's a relatively simple principle. Is conductance or resistance of the cavity means that as you increase the blood volume, the resistance goes down, and as the heart begins to eject or you reduce blood volumes, the resistance goes up. And this plots now as very high fidelity in terms of temporal and spatial domain, since you're using a current to really sample all parts of the ventricle. You get this time-varying resistance or conductance throughout the cardiac cycle. So how do we convert? How do we go from conductance or resistance to volumes? Again, it's a relatively simple calculation. You can see on the left here, you can see a cross-section of the heart. This is a multi-electrode conductance catheter. And essentially, the voltage drop, the current that is injected between the outer electrodes and the voltage drop that is collected between successive pairs of electrodes, those pairs essentially act like a cylindrical segment in the chamber. And if you have multiple segments, then all those cylindrical segments are added up to give you a final volume. But how we can actually do the conversion, it's really based on Ohm's law, as you can see here. Since the current that we're using in all of the conductance systems use a constant current source at different frequencies for some of them. But since the current is constant, then the voltage that we measure across a successive pair of electrodes is proportional to the resistance. And again, it's here based on Ohm's law. So you fix I, V is proportional to R. Now, if you look at, if we blow up one of these segments, again, we indicated that they're modeled as cylindrical discs, basically just stacked up inside the left ventricle. The resistance for each of these cylinders is, again, provided by electrical theory of a conductor, and it's related to the resistivity of the material inside that cylindrical disc. Could be blood, could be saline, and this is proportional, multiplied by the distance between those electrodes and the cross-sectional area of that segment. Now, the volume of this cylindrical segment is just simply L times A, and if we substitute from the above equation into this, we have the fundamental equation for converting a volume to a conductance. And you can see that volume here is proportional to the resistivity of that material, the distance between those electrodes squared divided by the resistance. And this hence conductance catheter, because volume is proportional to 1 over resistance, which is essentially conductance. And this equation actually, for historic purposes, was derived by somebody named Jan Nijboer back in the 50s. I know it's attributed to Jan Bond in the 80s, but if you look at their initial papers, they actually reference some of the early work of Nijboer back as late as 1959. So we have a relationship between volume and conductance, essentially. So why this discussion on why do we need to calibrate? And it turns out that it's really, as we indicate here, it's all about location, location, location. That previous equation was based on very simple assumptions of a completely cylindrical geometry and a uniform placement of the catheter, always in the center of those cylindrical discs. Obviously, as many of you are aware, it's sometimes it's very, very difficult placing the conductance catheter, no matter what animal model you're using, in a very consistent location, and hence this introduces variability in obtaining a precise correlation between volume and conductance. So the necessity of calibration comes from, again, the variability in the measurement and placement of the catheter itself, but also in the variability from animal to animal. The most important variable in contributing to this variability is placement of the catheter. The catheter ideally needs to be placed within the central axis of the left ventricle. And it's much easier to do this if you're doing an apical approach, for example, but when you're coming down through the carotid and across the aortic valve, it's relatively difficult to control where this catheter will be positioned. The other factor that can affect the translation from volume to conductance or conductance to volume is the composition of the myocardium. And we've seen particularly in mice while we were working, the different strains of mice can actually produce different values in terms of the conducting properties of the myocardium. And finally, when you apply calibration, I think what's most important to experimenters is you reduce a lot of the variability that's already present, the physiologic variability, but you can reduce the measurement variability so that we can reduce the number of animals that are needed to either show an effect or a lack of it. Now, in terms of calibrating the volume signal, there are essentially really 4 methods that have been used, even historically. One has been a relative common factor, and this was used as early as the 80s, where they applied an in vitro calibration, where they just put the conductance catheter in a plastic cylinder of known volumes. They matched the resistivity with saline to that of blood, or even used blood itself, and constructed a calibration curve based on different volumes of cylinders. The other technique, which is known as reference methods or gold standard methods, such as MRI, echocardiography, more recently the introduction of 3D echocardiography or contrast angiography, is particularly the last one, which has been used clinically for many, many years, is when you apply these techniques, you actually get measurements of end-diastolic volume and end-systolic volume, and then you calibrate your pressure volume loop based on that. So you actually have an absolute calibrated pressure volume loop using any of these 3 methods. The last 2 methods are typically applied, the first one to calibrate relative volumes using hypertonic saline, and the last one is stroke volume calibration to get the stroke volume or the width of the pressure volume loop using a flow probe or a thermodilution technique. Typically these are used together within an animal because they complement each other in the information that they provide. So before we go ahead and really talk about some of the nuances of the calibration techniques, I think it's important to realize, what are you trying to get out of your study? What assessments are you making? Perhaps you don't need to calibrate at all. Maybe just relative changes if you're studying the effect of a drug on the ESPBR, for example, or on relaxation properties of diastole. You may not need to calibrate at all, and just expressing relative changes within that given study may be sufficient. It's also important to realize that of the 2 parameters that we indicated before, calibration techniques of hypertonic saline and stroke volume calibration, virtually all parameters from the pressure volume analysis can be derived simply from the stroke volume calibration, without applying the hypertonic saline for absolute volume. If you want ejection fraction, then you would need to apply the hypertonic saline in addition to the stroke volume measurement. So let's briefly now talk about how do we actually calibrate the pressure volume loop. So the first, if we look at the panel here on the right, and we look at the width of the loop, which corresponds to the stroke volume, how do we go from relative changes in conductance, from end diastole to end systole, to now calibrate that in terms of an absolute stroke volume? And again, keeping in mind here that the resistivity of the blood we're assuming to stay relatively constant during the experiment, which is again, I think, a reasonable assumption. So the first method that we discussed was that of blood resistivity, or the cuvette methodology, which is again the simplest method of all the techniques. It's using a series of wells or cuvettes of known volume with a solution filled of known resistivity, which ideally matches blood resistivity. And the conductance catheter is placed in the center of each cuvette, and subsequently you can construct now a conductance versus a known volume curve for each of the volumes. You do a linear regression, and then this is now automatically applied within your software package so that all conductance values or relative volume units, depending on which software you're using, are now automatically converted to known volumes based on this calibration. It's important to keep in mind that when you're using this calibration technique, the implicit assumption is that the gain that you're applying here, the calibration factor, is relatively the same from animal to animal. The only slight variability would be how you place the catheter within the cuvettes. But otherwise, you're just assuming that this technique, that the calibration that you apply from these cuvettes, since they're identical blocks that you're using in each experiment, the same calibration values are being applied to all studies that you will be doing. Now, so we have calibrated values potentially based on the cuvette, and we indicated that the resistivity stays relatively constant, although not always. And kind of the quickest way of doing this is if you are introducing some solution in there that you think may affect the resistivity, it's always a good idea to monitor the pressure volume loops and see if they're moving back and forth or they're getting bigger for no apparent reason. This could be a sign that whatever drug you're using may actually alter the conductivity of the blood and may need to be adjusted for. But in addition to the cuvette, you may consider now adjusting that stroke volume or even verifying that the stroke volume that you're seeing is correct by actually measuring the stroke volume with another technique. A secondary measurement of stroke volume or cardiac output can be applied, and some of the most common techniques are the transit time ultrasound flow probe, which can be placed either on the ascending or the descending aorta, a thermodilution technique, which provides, again, injecting cold saline, or echocardiography, which can be applied to the either to the aortic root to get Doppler waveforms or across the heart to measure end diastolic and end systolic volumes. Clearly, this could be challenging when trying to do this in mice. And even though you may get values, it may introduce a larger source of variability. So the way you do this, essentially you now have your stroke volume, whatever you're measuring, like what's called here your Millar catheter in this case, and then you take your reference volume based on any of these techniques, the stroke volume that's provided by that. You divide the two and you come up with essentially a correction factor. And if your stroke volume happens to be pretty accurate compared to this reference value, then this value will obviously come up close to 1. But if it's not, like in this case, for example, it's almost 2/3 different, then you can now apply this correction factor to that stroke volume. And again, this is when you're doing this almost snapshot type measurements at steady state. You're assuming that there is a linear relationship between your stroke volume from your catheter and your stroke volume from your reference source. Now, another method, again, which is based on a reference source, but this is the one that typically we used. We did this in every mouse when we were doing our studies, and we used instead of a steady state stroke volume as a reference, we actually left the flow probe in place. In this case, it was a transit time flow probe, and it's really the only technique that you can apply this methodology to. But you leave the transit time flow probe in place, and then you do an IVC occlusion, and you look at what's happening to the stroke volume, both from your conductance catheter, which you can see here on top, and from your flow probe, which you can see here on the bottom, and you get stroke volume changes over several beats instead of just at steady state. And here, when you plot the stroke volume from the flow probe against the stroke volume from the catheter, you do a linear regression again if it's a reasonable linear fit, which in most cases it was in our studies. And then you come up with the slope of this line, which in this case was 0.15. And this 0.15 now will be your calibration factor that you can apply to correct the stroke volume measurements from your catheter. Now, there have been descriptions of nonlinear systems, like the admittance system, for example, where there could be nonlinear measurements between your stroke volume from the catheter and the stroke volume from the flow probe. But using this technology, and even if it's a nonlinear system, you're using the admittance system, it doesn't hurt to put on a flow probe and actually do a preload and assess the range over which your signal is actually linear. It could be bilinear, so that there could be a double correction. For example, at larger volumes, when you exceed certain volumes, you need to apply a certain calibration factor. And then over a lower volume range, there could be another calibration factor that needs to be applied. But they can be bilinear or even linear in most instances. But at least with this technique, you realize the full range of your system. And this technique will also verify that your conductance catheter is actually in the right spot to assess stroke volumes over large changes in left ventricular volumes. So it's a very, very robust technique, and it gives you immediate information on the range of your dynamic range of your system. Now, again, I indicated that we used to apply this in every animal. I know it would be convenient and very handy if we could just derive a correction factor from 10, 15, 20 mice or whatever, and just average them and apply this each time. We actually tried that and found in many cases, there was nearly a twofold range in the calibration correction values, and essentially we abandoned the technique of coming up with a single calibration factor. And it really goes back, I think, to what we discussed earlier, that it's extremely difficult to place the conductance catheter in the same spot each time. The myocardial properties, not only between strains of animals, but between diseased animals, normal hearts, hypertrophied hearts, heart failure hearts, which have thin walls, will greatly alter that calibration constant. So I think it's from my perspective, I think it's really, really important to try and obtain calibrations in every animal that is done. And again, the key to applying a lot of these factors is, again, you're assuming a linear fit in all the corrections between the stroke volume that you're measuring, whether it's a cuvette or a transit time flow probe. And you need to realize that when you're applying that calibration factor, if you're just taking a spot measurement, for example, that all the outputs from your PV loops, the most important outputs, as we discussed earlier, will be affected by this calibration factor. So I think it's important, if you can, to try and collect as many calibration measurements during each study. Now, we mentioned there was another two-step part to this calibration process, and the second part is what we call the offset, or the relative position of the pressure volume loop. So now, once you've fixed the width of that loop from your stroke volume calibration, you need to position it somewhere now on the volume axis. And typically, the loop shifts left when it's corrected properly, because, as we saw earlier, as we're injecting current into the left ventricle, some of this current leaks outside of the blood pool within the left ventricle. And the most important factor is the left ventricular myocardium. It can leak even further in larger animals over to the right side, the right ventricular blood pool or right ventricular muscle. And essentially, this contribution is what's causing the loop to be shifted further to the right than it should be. And this is what, when you're subtracting this extra volume, you're subtracting anything that's outside of the blood pool within the left ventricle. And this term is called the parallel conductance or parallel volume. We indicated the most common methodology is the hypertonic saline. Again, you can use echo and try and get an end-diastolic volume or systolic volume. But I think even going back 20, 30 years, this really turned out to be the preferred methodology. It was much less variable than the other techniques. And essentially, what you with the hypertonic saline is you inject a very small bolus of typically supersaturated saline solution, which is about 15–20%. And what we ended up doing was we had a 30 gauge or 31 gauge needle that we would insert in the jugular vein of the mouse. It doesn't necessarily have to be within the right ventricle. We got good values just injecting it in the jugular vein. We injected about 10 to 15 microliters, very rapidly, bolus injection, into the jugular vein, and then looked at the wash-in curve with the pressure volume loops. And they're pictured here on the left side. What you see is the loops will shift rightward and they will get bigger at the same time. And what this technique essentially does is, if you look at the graph on the right panel, the dotted line is the line of identity, and the other line with the red crosses there, or pluses, is taking the end diastolic volume and end systolic volume from each of those loops during the time that the saline is being washed in. And it regresses those loops until that line intersects the line of identity. And at that point, at the line of identity, you're essentially saying that your end diastolic volume and your end systolic volumes are equal. So theoretically, what you've done is, or mathematically, is you've rendered the blood non-conductive. So any signal you're getting delineated by that intersection point must be coming from outside the left ventricular blood pool. And that volume now you can use to subtract from your pressure volume loops, to center the loops now over to the left. It's also important to realize that the gain factor that you collected from your reference measures of stroke volume or from the cuvettes also need to be applied to this parallel conductance correction factor. So just doing parallel conductance is not sufficient to get sort of absolute volumes. This is again an example of what your pressure and volume signals look like during this saline bolus wash-in, and it's important to realize the key assumption when you're doing this technique, otherwise it invalidates the method, is that the hemodynamics need to be as stable as possible during the wash-in of the hypertonic saline, just like it does for any thermodilution technique. So that if you're injecting too much and you're increasing the stroke volume, that's not going to be a valid measurement. And you can see on top there the left ventricular pressure, which is a pretty good guide, or you can use DPDT to see if you're actually altering the loading conditions when you're injecting your saline. These need to be, the hemodynamics need to be absolutely stable while the saline is washing in, so that essentially all you're doing is altering the blood properties, the resistivity properties of the blood. Now we, again, in keeping with this parallel conductance calibration, we saw from the previous two slides the graph where we used the end systolic volume and the end diastolic volume. There is another method that was developed in Dave Cass's lab, published by Ed Langford in 1990, where they looked at not only the extremes of the volume curve, but they took the volume curve and divided that into 20 equal or X amount of isochrones from end diastole to end systole. And they've regressed each of those isochrones now back, as you can see in the bottom corner there. And theoretically, they should all regress and intersect at a common point, again being the parallel conductance. Now the reason this was done was, one, to assess how robust the technique was instead of just looking at 2 points in the cardiac cycle. Now you looked at multiple points, and this verifies that you've done a good saline calibration. But it also, more importantly, assesses if the parallel conductance is changing throughout the cardiac cycle. And if it is changing, then each of those regressions will not converge to a single point. What they essentially found in their paper was, I believe this was in canine hearts, was that there was very little change in the parallel conductance throughout the cardiac cycle. From my experience in mice, we also saw the same thing, that there was very, very little deviation of the parallel conductance throughout the cardiac cycle. And whether you use the extremes of the volume or this technology, it was, or this methodology, sorry, the estimate was quite robust. I know there have been other publications where they have shown evidence of time-varying parallel conductance throughout the cardiac cycle. But again, this needs to be applied in your own study within that given model that you're doing to validate or verify whether a single point measurement is sufficient, or you may need more complex methods of measuring the parallel conductance. And finally, sort of a methodology that is between those two, you can also plot the stroke volume against the total volumes that you measure and regress end diastolic and end systolic volume points. So this is a compromise between the two techniques, and ideally, again, those 2 points, if the parallel conductance is not changing and you've done a correct wash-in procedure, they should intersect at a common point. And again, if you look at the x-axis for the stroke volume, they come back to rendering the stroke volume 0, which again, theoretically renders the blood non-conductive. So once we have, again, fully calibrated data for stroke volume and for end diastolic volume, you now have the ability to measure the ejection fraction. So when you have both calibration techniques applied, you have all the other parameters that you can derive from the pressure volume loop. You now, in addition, have the ejection fraction, which again could be important measurement if you want to assess any remodeling changes. If absolute volume is important to you, then you need to apply both techniques. I would like to point out at this point, unlike taking multiple samples or measurements of a reference measure for stroke volume, doing repeated saline calibrations can be quite challenging, particularly as saline affects calcium cycling in the heart and is a pretty profound myocardial depressant. If you're looking at models where the animals are in heart failure and you've spent 2 months trying to develop this model, you may want to assess maybe just doing 1 at the most 2 measurements, or perhaps even not more than 1, because you could actually kill animals if the technique is not done right, if you've injected too much saline. So in some cases, you may not always be able to get repeated measurements of the parallel conductance, and typically, even in healthy animals, you probably can't get more than 3 measurements at a time that you can average. So as we're close to wrapping up, just a couple of quick points. As people spend an enormous amount of time, and these the calibration techniques are obviously quite challenging to do for each animal, but equally as important, once you have a fully calibrated pressure volume loop, it's important now that when it comes time to doing your analysis, it's equally as important to understand some of the techniques that are applied to this analysis, because that could introduce even more variability than perhaps not calibrating your system to begin with. Or if you've gone through the trouble of calibrating your system, you've now introduced so much variability by some of the analysis techniques that you've essentially nullified your calibrations. And just to quickly highlight an example here is if you look at, I'm sure many of you that are working with rats and mice, every time you do a preload reduction and construct the ESPVR, you can see it's very curvilinear. And as you're applying fits to this, if you're trying to apply a linear regression to a nonlinear ESPVR, depending on which loops you've actually picked, you can get very, very different slope values. You can get different, you can see from the linear relationships here, different values for your V0. This will affect your coupling ratios, as you can see here with the EA values. So it's important to realize that to understand the analysis techniques that you're applying, if you're applying a linear fit, you need to adjust your data that you've collected, the range you've collected, so that it is essentially a linear data fit. You cannot use nonlinear points and apply a linear regression to that, because it becomes extremely variable which points are really driving that fit. And similar, for example, if you're assessing tau values, a time constant of relaxation, which is a mono-exponential fit, if you look at your raw data points collected from the aortic valve closure to some value above end diastolic pressure, that curve needs to be mono-exponential. Otherwise, you introduce enormous amounts of variation. And there's a really great paper by Burkhoff, Mirsky, and Suga, going over some of the analysis techniques. And so it's important that when you go through the trouble of calibrating, that you apply the appropriate techniques so that you reduce your variability as much as possible. And the second point here comes from experience from several times that, once you pull out the conductance catheter, it never hurts to just put it back in saline and see what the pressure sensor is doing, that it should be close to zero again during your study. Otherwise, if your pressure is drifting during the experiment, this could be caused by the sensor could begin to fail, and in most cases they don't just fail immediately, but they fail and become very unreliable from study to study before they completely die out. It's always useful to just put the catheter back in and just verify that your pressure is close to atmosphere like it was before you placed it in the animal. So to summarize, being able to collect pressure volume loops and why I think people are still using them is really there is no more powerful technique that you can use to apply and understand exactly what has happened to your model or to any intervention that you've given. Separating preload, afterload effects, deriving diastolic, systolic properties, and coupling of the heart to the arterial system. But again, you may not need all of these values, and it's important beforehand to identify from your study design what exactly you want to get out of your pressure volume study. And then again, once you apply your calibrations, how will your analysis techniques now influence the data that you're going to get from this? So it's important that, again, you choose your calibration procedure accordingly. You try, whatever that is, you try to be consistent from animal to animal. You may not need to verify in every animal, but sometimes make spot measurements within an animal to make sure that your values seem reasonable. And I think in the end, for me at least, from my experience is a proper calibration should be your default procedure, and ideally should be done for each experiment.
So without further ado, let's move on to our Q&A session. So I think we're going to kick things off here. We've got some questions that have come in about the stroke volume correction factor. So I'd like to start there, Jim.
Sure.
Can you further explain the difference between the alpha stroke volume correction and the flow probe stroke volume correction? So this is going back to those slides, I think, where you were talking about a snapshot versus a transient stroke volume or time varying stroke volume calibration. Specifically, how are these different, and how importantly, how should a researcher choose one over the other? So just maybe your thoughts on that.
Sure. Sort of the alpha or the stroke volume, I mean, they're essentially doing the same thing. If you're going to derive your alpha from a cuvette, again, the cuvette will give you the same value each time. The only variability being the resistivity of the solution and the placement of the catheter. The stroke volume has two aspects to it. One is at steady state, using whatever reference measure you need, and one is during a preload occlusion. Theoretically, those values at steady state and during the preload occlusion should be the same. The conductance signal is inherently nonlinear, but if there is a linear range, so if you're within that range, then you're fine by applying the steady state. If you're outside that range, which you will now see by your IBC occlusion, then you may want to apply sort of a different correction factor based on that regression that you've done over beat by beat. But that's really the key difference, is you're able to identify during the IBC occlusion the range where your conductance catheter is linear, and you can apply a spot measurement.
Perfect. Okay, very good. And kind of continuing on the topic of calibration, what is your standard approach to choosing how to calibrate? So what do you do most often, why, and when have you chosen something different?
As I indicated, when we do these studies, if we're going to calibrate, then we do both parallel conductance and stroke volume calibrations for every animal. In cases, there have been cases where as I indicated, relative changes were sufficient, like in some of the devices that we're testing where we can turn the device on and off. We bypassed any calibrations and just use the relative changes. So typically we either calibrate fully or we just express it as relative changes.
Okay, so kind of working at the extremes is, in your experience, you tend to do. Okay, great. Folks, I'm also going to bring Brandon Boucher online to our Q&A. He is head of the research business unit at 80 Instruments. Hi, Brandon. Do we have you with us?
You do.
Excellent. Thanks for being with us. This question I think you'd like to chime in on. So specifically, Sarah Roldan has asked, in a multi-segment catheter with 10 electrodes, in which she needs to block some of the electrodes since they don't all fit inside the ventricle, should she block the first electrodes in the catheter or the last electrodes of the catheter, or both, or what's the prescribed kind of best practice here for when you're dealing with a multi-segment catheter?
Yeah, it's a good question. Essentially, the way you want to go about this is to look at first the way the catheter approaches the ventricle. So typically, with a multi-segment catheter, you're going to be going in through the aorta, and what that means is the most distal electrodes will be at the base of the ventricle. The ones proximal to you will be presumably outside the ventricle in this case. So what you want to do is, if some of those are outside the ventricle, you'll turn those electrodes off, moving the excitation electrodes closer to the ventricular space and ensuring the ones that are actually inside the ventricle are doing the sensing. So the answer to the question essentially is the electrodes that are outside the ventricle are the ones that are turned off, and the excitation moves closer to the space.
Perfect.
If I can just add something to that, this is we've actually run into the same issue, and one of the advantages, you can also plot, you can look at each of the segments, the individual segments. You want to keep as many segments as you can active, because you're trying to sample from the entire volume. But if you look at the segment that's outside the ventricle, its volume will be going in the opposite direction of the ones inside the ventricle. So when the ventricle is ejecting, you'll see all the segments inside the volume decreasing. The segment outside, its volume will be going up, and then you can identify which one to turn off.
That's a good tip. That's very good. And actually, I was going to say another question that has come in from Thomas Sharp. It's on the same note. So he's working in a pig ischemia reperfusion model, and the heart dilates over time. So he says, when using a catheter that has multiple conductance segments, should I use the same number of segments as I use in baseline, and I assume here he's suggesting with healthy animals, or include new segments which may not have been included in the initial measurement? So as the disease progresses and the ventricle enlarges, or maybe the converse in a different model would be it gets smaller, how should one adjust, I guess, their strategy as to what they're recording, with the segments?
Again, I would try to use as many segments as you can and be consistent using those segments over time. I think also the advantage of the conductance catheter, particularly in ischemic models where there's a portion of the wall that may be affected, it may be insightful to look at regional segmental volumes as well and plot pressure volume loops using that segment alone and track that over time, a consistent segment. I think that's also very, very informative.
Yes. And actually, in extension of this, I'm curious, so is it also correct that, each segment is set up as, or at least can be set up as an independent trace in your data acquisition software? So is it fair to say if segment 1, for instance, was, if it proved to be maybe a bit noisy or just you didn't want to include it in your final volume calibrations, that's something post hoc someone could decide. So is it fair to say record also as many and then look at the active segments you'd like to use for analysis post hoc, so that you don't turn something off and not have anything recorded from it, I guess is what I'm saying?
From my experience, yes.
Yeah.
And not only turn something off, but you can also record all segments, and you may see information in there that you wouldn't see by just adding the segments to get a composite signal.
Right, right. And Brandon, so obviously Power Lab can be set up in that fashion that I described, right? Where you have all your independent signals, you have a total, but you can also effectively post hoc adjust what the total volume represents, what segments basically make it up.
You certainly can, and some additional information to this is, yeah, you can certainly record from all the segments and gather that information, even if you make a decision ahead of time about how many segments you're going to be using, you can. But what you should also consider is that each of those will affect the way your calibration works in a multi-segment catheter. So if you're going to plan in advance to have, a certain number being used during one portion of the study, and perhaps you have to expand that, keep in mind that each of those have a volume associated with them. So you want to make sure you retain that information so you can apply it later.
Okay, great. Let's switch gears to talking about the saline injection. So, Jim, you did mention, there is some potential risk or effect of doing too many injections. Sarah has asked, should a saline injection be done at the beginning of an experiment or at the end? And then also, how might this change if someone is making measurements from the LV versus the right ventricle? I mean, is the technique the same, or I guess is the strategy that you presented, does it change slightly if someone's doing RV PV loops?
It shouldn't. It should be identical. It's a little more challenging on the right side because of the shape of the loops. But the theoretical concept is the same. In terms of at the beginning or the end, I've typically tried to do one as close to as possible as when I was making my critical measurements. That typically was more closer to the end of the study.
Right. I think in general it would be at the end of the study. Is that correct, Brandon?
Generally, yes.
Yeah.
It would be. There are only a couple of circumstances where I've seen people do it at the beginning out of necessity. But that's really, again, sort of the theme of this, and thinking ahead of time about how things are affected by your approach and what you're actually doing in your study, and if you need to change that, you can. But typically, yes, we do it at the end.
Okay, perfect. How about your experience with the addition of heparin to a blood solution when you're doing a cuvette calibration? Is this a suggested best practice? And if people are using heparin, should they expect that this changes their calibration, so to speak? Does that have any effect on blood resistivity and the resulting cuvette cal that comes out?
Yeah, I don't know if Jim has any thoughts on this too, but essentially the answer is yes. We do recommend that you use heparin when you do the cuvette calibration. And that's just to prevent clotting, and what we essentially recommend is that you minimize the amount you use. Typically what we'll coach people through is to coat the syringe you're going to be capturing the blood with. And so you have a very small volume of heparin being added to the blood pool. And find out how that works for you, because a piece of this is, to try and do this quickly so that the blood doesn't lose any temperature and doesn't have the clotting associated. So you're trying to, stem the time that it takes by adding a little bit of heparin, and at those small volumes, we haven't seen that effect. But yeah, do keep in mind that heparin at larger volumes will affect the resistivity of the blood. So to mitigate that, we recommend using a minimal amount and make sure you do it quickly so you have a balance of both situations there.
Very good.
Yeah,
Jim?
I agree. And if you're using a reference standard, if you're measuring flow anyways, then this will not. The heparin won't affect that alpha calibration once you've set your stroke volume from the flow probe.
Okay. Very good. All right, question from Mark Kearns. He states that, so he often obtains very clean-looking hypertonic saline calibration, so good for you, Mark. So much like the ones presented. However, he finds a big challenge in selecting specific waveforms for the parallel volume adjustment, so basically working within software. The variability, depending on which waveforms you select, so this is his experience, can be quite large. So, Jim, how do you approach looking at a saline wash and making those selections so it's consistent and in your eyes accurate from animal to animal?
You should only select loops where you can identify where the saline is beginning, and then again, you don't need that many. You may not want to include, let's say you've got 10 loops. You may just need, the first 5 or 6 of the saline wash period. But it's important to exclude sort of the steady state loops and just start where you see the first deflection in that waveform. And you can do, again, you can look at, if you want to verify, you can look at multiple those couple techniques that we discussed in assessing which data range to pick so that they all converge down to one consistent value.
Right, and that paper is referenced. So folks, afterwards, when you're looking for this content on InsightScientific.com, you'll have a link within the slide deck to get to that paper that Jim noted in the middle of the presentation. Very good. Okay, just give me a moment here. We've had lots of activity in the Q&A, kind of in the submission panel here, and I'm going to just sort through some of them. Okay, let's talk about if someone is looking for, if their goal is to determine small differences between mice in cardiac function, and they want to look, they like stroke volume is an important measure for them. What would you recommend as far as a calibration approach, which is going to be best serving to them when they're looking at small differences in mice? And again, stroke volume is a key parameter that they want to report.
Sure. I would say for me, what I would consider the gold standard is a transit time flow probe.
Okay. So transit time flow probe, thus giving you the ability to look into some of the time varying calibrations that you also talked about, Jim, and applying like a full absolute volume calibration for their PV data?
The transit time will give you a stroke volume. You may not need the actual, if you want the absolute volumes of the ventricle, you would need a hypertonic saline, but the transit time alone will give you the cardiac output and the absolute stroke volumes.
I think in this case, they certainly want the PV loops to, as an independent, so transit time stroke volume, and then use that throughout the procedure, but also use it as a calibration reference for your PV loops, and do the full absolute volume calibration as you discuss it. Okay, very good. John Kingma has asked, do results, pressure volume loop results differ when the approach changes? So two most common approaches, I think, and we'll just use the mouse in this situation, is they're talking about an open chest approach, apical stab, or going retrograde through the aortic valve. So in your experience, Jim, does the approach kind of change the way you maybe look at doing analysis, and do you see any just common differences between the way that the data is recorded?
That's an excellent question, and we struggle with this a lot. Most of the work that we published was with, and it was open chest in the sense that we did a limited thoracotomy. We don't have to open the full sternum to get the catheter in, so a limited thoracotomy near the apex. We found that was the preferred method because we could control where we could place the conductance catheter versus placing it down the carotid and going across the valve, and then we were quite limited where the catheter sat. And part of this is not only the calibrations, but for some of the disease models, it's important to get a relatively nice-looking PV loop that you can actually calibrate and then subsequently use that for analysis. If the loop is sort of twisted on itself, or if it's sheared to one direction, where's your insystolic volume if it's sort of tilting sideways? Is it at the top left, or is it at the bottom left? So trying to get a loop that's as box-shaped as you can, so you can apply your calibrations in your analysis, I think is important. And so we preferentially use the apical approach with a minimal thoracotomy between the ribs. Okay.
Very good. All right. Another question, moving on to load-independent measurements. Jim, in your experience, what are your kind of best practices and tips for fitting end-systolic pressure-volume relationship? On the last slide, you talked about how there can be extreme variability, and people should also consider whether they're going to be doing linear or curvilinear fits. So again, are there kind of rules of thumb that you prescribe to ensure that the measurement is, again, accurate in your eyes and also consistent from animal to animal?
Again, a very good question. We took two approaches to this. We defined, if we were going to do just a linear fit, we defined a linear portion, let's say between 80 and 100 or 110, and applied that range to all the animals. It can be challenging in heart failure mice where that range may not apply, but in most cases you typically can get a reasonable range that you can use across all animals and then fit your relationship to that. Mm-hmm. The other, if you want to use the more curvilinear logarithmic approach, I think that paper I mentioned by Burkhoff, Mirsky, and Suga described there are techniques that are published where you can actually fit parabolic or logarithmic fits, mm-hmm. and then you wouldn't so much extract one number, but you would use that entire regression and use that entire relationship now to compare from animal to animal, and that's fairly robust. In it also, you can also transform your data. You can do a linear transformation, almost like a stress-strain analysis, and linearize now offline the logarithmic curve and apply, and those relations are very linear. And you can apply fits to that linear regression.
Okay. Very good. That's a great answer. I mean, this is something that I think is going to come up, and there's not really a solid, black and white answer to how to fit curves in a lot of PV science. It's not a one-stop shop type of thing. So, but that feedback is excellent, and I think it's very helpful.
And I think it's at that point when it comes to curve fitting, I mean it's not physiology anymore. It becomes mathematics and statistics. So, make your statistician or mathematician or engineer your best buddy, and yes. help you figure out some of these fits. Never hurts.
Yeah, I was actually to say that's a good piece of advice. I have heard many say, even if following all the best practices at the benchtop and all the best practices when working on the data analysis software, and they've kept to a gospel, so to speak, and they're very confident. And now it's, what do I do with these numbers, and how do I properly evaluate them statistically? And that often that opens up a whole other, bucket of worms. So
it's a whole webinar.
Yeah, exactly. So yeah, seek out experts. That's going to help you present your data properly for publication, right? So really good piece of advice. Final question here. We've had a couple people ask about studying multiple species. There was also this note about multi-segment catheters. Brandon, can you just clarify for the audience how is the Molar-80 instrument solution kind of presented so that you can cover a range of animals, either even as small as mouse, but going into medium size and large animals? If someone's trying to set up their lab or make sure that they cover all ends of spectrum, what do you guys offer for them?
Yeah, we have solutions for all of the commonly used and not commonly used animal models. So we start off with a solution that's really based around the rodent-sized animal, and that provides simply the single segment that you saw in some of the earlier slides, the capability to do that. So a single excitation pair and a single measurement pair. Mm-hmm. That's a single segment solution. You can buy the catheters that work with those rodents that are appropriately sized for the animals you're using. And with the same hardware, with some added capability provided with that hardware, you can also get the multi-segment catheters that work with the slightly larger animals in sort of the rabbit-sized area.
Okay.
But you're using the same hardware, just a slightly different software capability to expand the capabilities there. And then again, same recording hardware and a different catheter with multi-segments that is larger for the larger animal models.
Okay.
So really working with similar hardware, different catheter options, and expanded capability for those multi-segment catheters.
Okay, perfect. So just to clarify, effectively one system has the ability to cover all animal ranges between single segment operation and multi-segment, and if someone invested in one or the other, they could upgrade, so to speak.
Yeah, it certainly does, and you could certainly purchase one system and different catheters that'll approach your different animal species.
Mm-hmm.
So you can have one system on the benchtop with the actual catheters you're interchanging for different animal models. Keeping in mind that on the back end with our LabChart software and what we provide there also has the capability to cover the full range in PV loops, but also really any other work that you might be doing in your lab as well.